And I'm Mohit Mazumdar, and we will start today's session now. So we'll start as usual with our discussing our group progress. So last few days, after our session on 10th, we had some spikes in both server and educational access, and people, especially Shayok, did some pipelines. He ran a few pipelines and also did some courses. And after that, we did not did on February 13th we postponed that class to today because of several reasons, one of which is to look into the material that we have already covered so far. So we have covered a substantial bit of transcriptomics series, transcriptomics 1 and 2 in depth, and then we moved into transcriptomics 3, which includes the machine learning approaches. And in the last class, we discussed several machine learning approaches and why people are using those machine learning approaches, plus how to understand which one is which. Like, very simplistic question would be to understand the basic of which machine learning is used for what kind of data, and also what different kinds of algorithms are there within those different approaches, like supervised machine learning method and unsupervised machine learning method. We learn those how they work and what kind of data they take. So today, we will again revisit some of the concepts, and again we will make understanding that how this whole thing is working in the research projects that are ongoing nowadays. So if you see the kind of research which we keep on sharing on our LinkedIn, Facebook, and also individually, like Soham, I know, shares a lot about the current state of research that is ongoing. So he's been doing it very actively, and I'm really proud about the fact that he's doing that. And because it helps us to learn new things, just to going through those articles or small blogs where describes the current research. So it also helps us to have a broader perspective in for our future, to which area we would like to work on. Because as I see, and as things are progressing, we are moving towards era where analysis would play a big role, because it will be easier and easier to obtain data, and then it would require scientists to understand the data and then design experiments according to that data, according to that information, according to the knowledge that we have already gathered. So bioinformatics in general plays a important role in that aspect, because we are looking at every sort of data before going into the research, before we design something, or before we trying to learn something. We want to know that what has already been happened. So that is pretty important. And to understand what has already been happened, first of all, we review the publications. We see that what is out there, what has been published, what has been validated, and whether the publication that we see is also, like, how relevant it is. So that sort of basic understanding is pretty important for us, for you to do better in your career. So right now, as you're going through your different programs, mostly bachelor's programs, we focus on the things to learn about the basics, and we focus on the things that are there described in our syllabus. And then we think that if we are covering this, why to sometimes we don't bother to do more, because we think that we are already being having so much load of doing all of which is required to do and to finish the degree. But it's just kind of an awareness that is required. We don't ask you to, I mean, individually saying, I don't ask you to, give all of your time to the things that are happening all around the globe, but to at least give 5 minutes, 10 minutes of your time in a regular way. That will help you in a long, long way, because over the years, when you collect this information and you happen to have a discussion, you will be knowing so much more. So in that context, I would like to talk more about so I see there is a question maybe by Arundhati. So Arundhati, can you unmute yourself and ask, or is there something in the chat? Let me see. Yeah, thank you, Beepsha. So recently, I mean, yeah, I was doing some sort of, seeing the kind of scenario is in India about the bioinformatics growth, because as I was talking to, as I was talking to some of the students, also we got to know about that key in India, bioinformatics is growing because of the government's initiatives, and also because of some of the institutes that have been in there and been doing for a long time, but now, I mean, they have evolved, which we were not aware of. So I did an analysis, I did some research, I read a few articles, and this is the article that Deepsha has shared, that I came up with, and we have published it in using this URL. So if I open this, you will see the article actually describes the current scenario in in India. So big data bioinformatics education in India, and how can we stay competitive. So it also describes the things that are there, that the initiatives that India has taken for bioinformatics research over the years, and also the current state of universities that offers several courses and educational bodies, and how, where they are located, things like that. And this graph especially shows that that India came to the fourth place. I mean, that's not at all bad, that's really good. So here, if you see, mm, so last year, India was ranked fourth among the top 20 bioinformatics database contributors in the world. So that is really a good achievement, I believe. Coming after, we ahead of us are US, China, and UK. So there's a lot of bioinformatics contribution that is happening. So this article describes all that. This article describes what we are offering, and also our collaboration with BioClues, which is a very good organization and helps students to in their research. They help their uh, they don't charge you for anything, because they are a academic institution, and you can learn a lot about the ongoing research in India. A group of bioinformaticians have started this, especially Prashant here, who is an amazing scientist himself, who's working in Jaipur, in the department of Birla Institute of Scientific Research. So he started this group, and they are doing really well in helping students. So when you have time, just browse through this article. I think it will be helpful. And also, Chandrani, this is the article I thought that we would work together. So maybe next time we will be working on some other article. Okay, so last time what we did was something like really interesting. We interacted via quiz. We talked about how we can answer those quizzes that are there in the course. So to make you more comfortable with the quizzes, and the reason of these quizzes is to help us understand that how well acquainted you are with the pipelines that we discussed, and also perform your own analysis. So I'm going to ask you a few questions, and let everyone provide their answers in the chat. So the reason of this exercise is that it will help us bring up any questions or clarification needed to understand how to use the methods we are learning about. So let's do this. The first quiz. So in general, it's this quiz is from transcriptomics 2, I guess, but it's a very general question. So I would want you to answer in the chat, and don't worry if you have the right answer or not. Just based on the whatever understanding you have, you can just guess. So we are not looking into any sort of marking or anything like that. So please feel free to write any answer you like, or you think is the correct one. So in general, researchers don't use t-test in RNA-seq research. Which of the following is a reason for this? First, sample sizes are too large, and with such a huge number of genes, t-tests get overwhelmed. Sample sizes are too small. The strength of RNA-seq stems from the huge number of genes analyzed, but often only a few samples are used for analysis. Is this correct? Or three or fourth? So three is t-tests are not suitable for hypothesis-free research. A common approach is whole transcriptomics. RNA-seq or t-test can only be used for categorical data. Can anyone tell me? If not, then I'm going to tell you the answer anyways. Option 2. So Soham is saying option 2. Okay. Anyone else? Okay, so the correct answer is in fact uh, B, which is 2. So it's because t-test is prone to noise originating from sequence depth and batch effects, and maybe easily influence the if the sample size is small. So if the sample sizes are small, t-test is not a test that you would recommend. So right answer, so. So let's jump into the second one. So which of the following paint the closest picture of your differentially expression results, so the differentially expressed genes? So the analysis started out with about 10,000 genes. We are talking about the analysis we did, which we learned through our sessions, and also in transcriptomics 1 and 2 you saw. So based on that, tell me the right answer. So the analysis started out with about 10,000 genes and ended up finding 100 that are significantly differentially expressed. The analysis started out with just over 50 and ended up with less than 10. The analysis started out with 5,000 and ended up finding about 1,000. Fourth is the analysis started with 100,000 and ended up finding less than 200 significant genes. So can anyone tell me in transcriptomics 2, from how many genes we came to how many genes after doing all the cleaning analysis and differentially expressed genes, removing those which are not important and all those things? What was the number? Does anyone remember? So how many genes we started with, and after cleaning up, how many we got after running DSEC and all? So the correct answer is, Soham is saying 5,000 to 1,000. Okay. Anyone else? Shinjini is saying C. Same option. Oh, okay. Soham is saying option 2. Soham, option 2 is 50 to 10. And you're writing 5,000 to 1.
No, sir. The option two is actually the for the previous question.
Oh, okay. Sorry. I got confused.
Yeah, yes,
So
sir.
you're saying option three, right?
Yes, sir.
And Shinjini is also saying option three. Okay. So the right answer is option four, not option three, because we had samples from number of mice. So there were so many samples from different patients. So we had like 52 or something samples. So all of those people have their genes in there, and that number of genes are more than 100,000, more than 100,000 genes that we started up with. And then after standardizing and normalizing, doing all those things, quantification, and then differentially expressed genes, we came down to 200, which is quite a good number in itself. So that's the correct answer. So yeah, the next question: which of the following can you say based on this PCA plot? So if you can see the plot, look closely, look at the options also. The transcriptome of normal like and basal cluster together. So you can see the dots. The transcriptome of all four subtypes overlap. The four cancer subtypes are differentiated from one another to some degree with a clear difference between luminal and the others. The four cancer subtypes are all clearly differentiated from each other on the basis of their transcriptome. And I think we saw similar in our analysis when we did four subtypes and we saw the PCA. So based on the dots, you can actually answer this one. If you see the the figure, then try to answer.
Sir, may I try to answer?
Sure,
Uh,
everyone is
so
invited.
yes, so in the graph I can see there are four plots and there are four subtypes. So in the first quadrant, I can see the normal subtypes and basal and yes, some clotting lower there. Now in the second and fourth, there are largely luminal, and in the third quadrant there are basal and clotting low. So I guess the option should be either three or four, but in three it is saying that the four cancer subtypes are differentiated from one another to some degree with a clear difference between luminal and all others. So I think the option is three, because luminal is just in one side of the plot and the other subtypes are in the other hand.
Yes, Richishmita says option three. Good job, Richishmita, and also Soham. The correct answer is indeed option three. That's nice. Okay, now which of the following would be useful comparison in differential gene expression studies? Useful comparison. So is it like, there are two also options which says all of the above, none of the above. So what do you think? When you want to see differentially expressed genes, which means that you want to see difference, right? Okay, Soham is saying Claudin low versus normal, which is option two. So option two, okay. One answer, option two. Option one. Chinjini is saying option one. Okay, I will tell the answer anyways, but I'm looking for more guesses or more uh, logical guesses. Okay, Richishmita is also saying option one. So luminal versus non-luminal. Okay, luminal versus non-luminal totally makes sense. So
There's like a
obviously A.
clear distinction between luminal and non-luminal, whereas in the other, including low and normal, there is overlapping of the.
Okay, there is overlapping. So if you have to, like, luminal versus nominal, non-luminal is correct. That's a correct answer. But Claudin law versus normal, like, is also could be a correct answer if you want to differentiate those two. I mean, in this graph I can see that of all of them are somehow being separated into different areas. So they, out of the distribution where everything was cluttered and everything was all around the place, we came to this graph where they are separated quite well. So I think the right answer would be all of the above, because we can even see basal versus normal, like, and we can do the separation based on there as well. So I think the correct answer should be all of the above. But yeah, luminal versus non-luminal is the most is the question, based on the question also, but it's the most obvious answer. So next question is, what kind of data is available from the cell line study we used in this course? So I went through a slide. In that slide, I said about these things. So I don't think all of you remember, but I mean, any guess? Although we have shared this link several times that to see this publication, but I think it's a big publication. Any guesses? I think RNA-seq is obviously we used RNA-seq, and and then I did talk about GI50 value, because here this publication they did several things. They did the RNA-seq. They saw the drug efficiency on different cell lines, that how this drug is sort of behaving against the different breast cancer. So they looked into that also. Also chemotherapy drugs also they checked. So drug efficacy is also there. I mean, it's obviously there. And protein abundance, they also did that. So all of the above. Right answer, Soham. Okay, I think this is the last question. Okay, so try this one also. Which might be one interpretation of PCA result of gene expression table. I also put a sample here. So after doing PCA, we got something, right? We got those things. So that is the question and also the answer. So what we got? Each component accounts equally for the genetic variation in a sample. Accounts equally. Okay, first is accounts equally. The second option is the first component accounts for 100% of the genetic variation. Okay. Third is the first component accounts for the majority of genetic variation in the sample, while the rest are insignificant. Okay. The first component account for the majority of genetic variation within the sample, while successive components account for the next largest degree of variation. PC1 is greater than PC2 is than PC3 and PCN. So tell me. Come on, last question, guys. Just guess. Okay, so option 4, Shinjini option 4. Oh, sorry, Shritama option 3. And Richishmita is also saying option 3. So when we did PCA, we got a plot and we saw those you know plots in three things. We looked into we we got we the option we gave was to get three dimensions. So we put PC1, PC2, PC3. We compared PC1 with PC2, we compared PC1 with PC3, and then there was a comparison between PC2 and PC3. But when we do PCA, we get PC1 to PCN, like based on the number of samples we have, because we were putting samples. So PC1, what does PC1 signifies? Is PC1 signifies the largest amount of variance. So the third answer is the right answer, not the right answer. Half is the right, half is right. So the first component accounts for the majority of genetic variation. That is correct. While the rest are insignificant, that is not correct, because we saw in that PCA plot that the amount of variation that we are capturing from that plot is like from PC1 we see 53%, PC2 we saw 17%, so 70% in total. So 53 and plus 17 is like 70%, but 30% is again there. So we cannot just say that it's insignificant. It's just that we are seeing at PC1 and PC2, and then in PC3 we are looking for more variations. But majority of variation is obviously in PC1 and PC2, but others are also important. Not that important, but they are important, but not insignificant. If we say insignificant, that means like there is no significance at all, but that is not correct. So the fourth option is the correct one, as suggested by many of you. So well done. And now this is over. Now we'll move to the session. So these clinical subtypes we talked about. So we have, like in biology there are so many subtypes, but in our data set there were only 4. So we have like 5 normal likes and 6 Claudine low samples in biology. So you can do your own research on the internet and find more about these cell lines. But the cell even look differently, as you see in this picture. So what we all know about the clinical classification is that Claudine low is more aggressive cancer, uh, and uh, and is often grouped under triple negative breast cancer. So we did this before, triple negative mm, and ER positive in our previous data set. So this basal, Claudine low, and these are more dangerous, or maybe more aggressive, is because they don't have that mm, biomarker, or they don't have that receptor on the surface which can be targeted for drug. So we learned that also. So this is that example that talks about it. But I mean, to have more basic understanding of how these things are, I mean, what is a breast cancer, how does it happens, and what we look into. So let's do some basic understanding. So what you see in this picture is a mammogram. Okay, so a mammogram, what is a mammogram? Mammogram is a x-ray picture. Okay, so x-ray picture of the breast that doctors used to look for early signs of breast cancer. So in this arrow you can see that there is something that is there in the picture which is not right. So this is a tumor which has been highlighted. So there are different types of tumors. Okay. So based on their location and all. So if it is inside, if it is inside, a lobular tissue, it could be uh, classified as a different cancer and henceforth. So as you see in this example, ductal carcinoma in situ means inside, lobular carcinoma in situ again inside, and invasive ductal carcinoma means it's outside, and invasive lobular carcinoma means that it is lobular but it is outside. So obviously, I mean, whichever ones are outside are more dangerous, because they spread, the possibility of them spreading is very high compared to the ones which are inside. So that is one thing. So once the tumor is found, once you see from the image, or maybe this is like the best example where you can see from the image, but in reality it's not like this much clear all the time. So we have several tests to understand whether whether what kind of cancer subtypes is there, and based on that subtyping, diagnosis, in a way you will be prescribed drugs. So it's very important to classify them in a proper way. It's like a kind of diagnosis. And to do that, I mean, what we have done so far is like these sort of immunohistochemistry experiments, where you see that based on the sample, yeah, and based on their straining, we can maybe classify them, whether what kind of tissue they have, whether it's like triple negative. So there's a pattern there which we have learned through experiments, and we know that if this pattern is there, this is this sort of luminal A, or triple negative, or HER2, or luminal B, what kind of breast cancer it is. So this is one way of doing it, which is like an immunohistochemistry experiment, which on a selective biomarker. So these you can check for these HER2 and EGFR, and these are BRCA1. So these sort of proteins we check, and these are biomarkers for looking into breast cancer. Another way is molecular subtyping, which is looking into a whole set of genes. So what we do, what scientists have done, is that they have created these panels. So this panel is called PAM 50 panel. So in here there are 50 genes, and what they do is to check the expression of this 50 genes in in the patient, the samples that they want to test. So what is happening here is that that the we know that in normal the gene expression we know, and also if these 50 genes are one of the cause of breast cancer, then from the expression level, as you know, green is signifies higher expression, and red signifies lower expression. So the more darker it is, so lower the expression it is. So more in green, it's based on the brightness and contrast that the expression is. So based on this pattern, you can actually see whether what kind of breast cancer it is, because luminal A is different from normal like, is different from luminal B, and HER2 and basal like. So here, I mean, also there are 10 to 12 genes which are housekeeping genes. So I think you guys know about housekeeping genes. So housekeeping genes are those genes which do normal cell function, and whether they are in a cancerous patient or whether they are in normal, they have the same sort of gene expression. So there are like in a kind of control to see that your experiment was whether this panel worked or not. Because in if it is to work, then the expression of those housekeeping genes will stay normal in all the cases. Like there will be no differentially high differential expression in those genes. So this is the molecular subtyping to understand the breast cancer. And also this is what we were talking about in the last session, And so there are several things that doctors do. So rate of survival and survival prediction based on this sort of cancer subtyping. As I was telling you, one is more aggressive, one is difficult to detect and prognosis. So based on this, there is like profiling survival predictions that plots are there based on the subtyping. So a couple of these sort of panels are available. These are clinical panels. One is PAM50, another is Oncotype DX. So based on like this is like a years of data that they have published, which shows that what kind of survival rate is there, and recurrence is there or not, stuff like that. So once you identify those things, then there are several ways to treat cancer. So one is like removing the cancer using surgery. Another is radiotherapy, which is another popular way, and mostly chemotherapy is also there, which is used. So what is happening in targeted cancer therapy is that based on molecular profiling, what we are doing is that based on your gene expression, we are dividing you into different groups, and then those groups are you know given drugs based on their profile, which kind of subtyping they have, or which kind of. So that is something sort of a targeted therapy, because what you're doing is not giving one drug for every cancer, but what you're trying to do is to differentiate between different cancers, and then providing them with the kind of drug which is more effective for one type of cancer and might not be effective for other type of cancer. So this is the publication that we looked into, and it's one of the way which we were doing it is to learn from the publication, right? So we what we did is that we tried to look into the data from the publication, and then we tried to do our own analysis on the server. So this is the publication, and when we kind of searched the data set, we found it in a bio project. So this bio project here, this is the link in here. So it's just to provide. Yeah. Okay, so as I was telling you about this bio project, so if you go to this link, and you will find the data set that we are using. So these data sets, what I mean to say is that these data sets are provided free, provided free to do analysis. It's just that we have to think about a problem, and then we have to start doing, such things, look for data, and maybe interpret. And then you have the T-Bioinfo platform where you can upload the data and run the analysis and do such experiments. And maybe in the future, I mean, many of you would be, doing some great work in this area. So, I mean, we covered a bit of the data set, and I think I will just stop here. That if you have any sort of, questions till now, and did you understand the this therapy, which is targeted therapy? So was it clear? That's what I'm asking. Like, till now, is it clear? Okay, great. Thank you. Thank you for telling. So if we do a review, what we did, we first processed the data. We did pre-processing from the raw reads. Then we did mapping. Then we did quantification. This was in transcriptomics too. And then we did descriptive statistics. We did visualization, and also Second, please. We did normalization. So this was part of transcriptomics 2, right? And then we start doing another part of hypothesis testing. We talked about the t-test and why we do, like, just like a biological question that, okay, this is my hypothesis, how to test it, whether I'm correct or not. So we looked into the statistics behind that, how do we do hypothesis testing. And then we did what? Data mining. We did PCA and hierarchical clustering and learned about data mining. And then we did classification, where we used, these machine learning methods, and see that how we can divide the data sets and then do predictive analysis, which is comes under classification. So interpretation is what? Interpretation, today's class was all about interpretation. So interpretation is what? After we get number of genes from the classification, we try to understand what those genes do, how can we annotate them, and how can we understand that how they are affecting the biology, why these genes are important, which pathways they are in, and what kind of processes they are affecting, whether they are affecting someone's metabolism, whether they are affecting something in the in the brain, whether they are affecting something in any of the other cellular pathways that these genes are involved in. So we do those things in interpretation for understanding better. So again, we did stop for the Q&A, but I think I'll just move on. That last time we learned about machine learning methods and how to classify. So what we did is that we learned that supervised machine learning take input as train and test data set, right? So let's simplify it further, and let's look at with one example which I was telling you verbally last time, and those are like the cute examples. What do you see in the picture are images, right? So we are seeing an example of unsupervised machine learning. And here, what we are trying to see is that we see a lot of images of animals, but as a machine would not know which kind of images they are unless it does some sort of analysis by looking at it. So once it is done, we can understand if we label them that, okay, machine, this is cat, this is dog, this is dog. And this is a part of the data set that we get before putting these images without any label, which is what we get in our unsupervised machine learning, where we don't know about the data. And then what happens is that we get data into different groups by doing what? By doing PCA, by doing hierarchical clustering and those things. What we do is that we divide them into different groups based on their features. So we divided the groups into cats and dogs. So this is what we get from unsupervised machine learning. And also if some feature which is like is an outlier, which is neither a cat nor a dog, so those features or those genes will come out as outliers because they are not being in a group which belongs to a particular pattern. What happens in supervised machine learning method? In supervised machine learning method, we are trying to do a predictive analysis. So we know from the unsupervised machine learning method that that we have a group of cat and dogs in a data set. Now what we did is that we build a training data set of both cat and dogs. So this data set will have both of them, so that the machine learns about both cat and dog. And what is happening here? We put something of a test for the machine. So test data set will also have cats and dog, but without label. So here the machine will be trying to, guessing or predicting which is cat and which images of cat and which images are of dog. So this is supervised machine learning method. So we learned about several examples of supervised machine learning method, but in each of these examples, if you see 1, 2, 3, the input data is the same. So we do what? Train data. Train data is what? Train data is with information. So pictures plus description, pictures plus description, pictures plus description, and test data is only pictures, because where we want to test whether it can predict the description or not. So what happens in decision tree? Decision trees are also machine learning, so supervised machine learning methods. So based on a threshold, it predicts the outcome. And based what happens in random forest? Random forest is a lot of decision trees. So it is again based on threshold, and what it does, it does prediction, which gives the output. SVM we learned, and LDA we learned, where SVM is based on decision boundary. So based on decision boundary, it does the prediction. So all these examples are examples of supervised analysis, where we do predictive analysis based on train and test data set. As I was telling you that we have last time discussed the train and the test data set, and I showed you that some the train data set has the Basel, Claudine Low labeled there, and the test data set does not have that information in there. So based on that, we judge the kind of prediction, and we see that how well the classifier has been able to predict stuff. So in our server, we did went into the data mining section, and there are two sections, which one is supervised, another is unsupervised. So we did both, and today maybe we will, if we have time, so we can do again this analysis. So as I was telling you, for supervised, we need test and train data sets, train with label, test with no labels, and usually we keep 70-30 ratio. So obviously the trace data set, the train data set should be always a bigger data set than a test data set, because you want your machine to have a lot of patterns analyzed, because next what it will be doing is doing predictions, which is very critical. So last time what we did is we did decision trees, and what we learned from decision trees is that decision trees are nothing, are very simple machine learning, is a very simple machine learning tool. What it does is that it just finds out the gene with which you can differentiate between two or three categories. So what it does is that it makes a rule that, okay, if this threshold, 0.67 threshold, is there, then whether it will fall into this category or not. So we looked at this example, and we learned that in the first graph what is happening is that whatever gene expression which is above 4.25 is luminal, and which and below that it would be any of the other three. Another example we completely saw showed us that for uh, not luminal, we are done with luminal. Second option is what we are looking at, the other three, how to classify other three. So what will this decision tree will do is that it will find another gene. So it will find another gene which will have such threshold. So it found out that basal has this gene expression in this gene which can help it classify into this sort of rule, which is 5.2. If in one gene it found out that basal has a higher expression which is above 5.2, and claudin low has a lower expression. So anything above 5.2 is in that in there is an important gene. In that gene, it shows that anything above 5.2 is actually basal. So based on that rule, it again formed a tree. So what it did is that last time we also talked that how decision trees actually work is that they do a random initialization. The algorithm works like that, that they started randomly from anywhere in your data. So they start and they will give you an output like this with like maybe three genes you will get, which will tell you that okay, you can classify your data set by doing like this. So we plotted these two genes, and then we saw that based on this information, our data set can be classified into several like this, where we have been able to see that okay, this is the gene expression. So what it means is that maybe these genes are important. Why these are important? Because they are the they are the thresholds for looking at the differentially gene expressed genes. So there is a distinction in that gene that is helping it to be becoming the gene that can do the classification, because it's an important gene, because it has a differentially expressed gene which is relevant. So what we will do in the next step is to learn more about this gene, which pathway it is from, and what it does. So based on that, we can connect the dots and understand what kind of role this gene play in the cancer and henceforth. So this is a data driven research. So another example, and this is another example, but it also signifies the problems, or maybe signifies the challenges associated with machine learning. So what we are showing you here is like a really optimal case where we have done this analysis for you, and we saw the best result and showing you like this. But when you do real research, the results are not so good, and like it will be more challenging, because you will not get maybe clear distinction. Then you have to do a lot of tweaking and twinning, and that is why research take time, and you have to keep on doing and learning, and changing a few things here and there to improve your result. So when we talked about 7,000 genes, then we saw that the classification is not so great. So we cannot, like, there are so many outliers yet. So what we did to overcome that is that running different random forests all the time. So maybe we will run 5,000 decision trees and random initialization, sorry, decision trees, and we will run different, like several decision trees, not one, because it is randomly initialized. So we will be initializing it over and over again. So what will happen? This will happen. So what happens? Random forest of decision trees. So what is random forest algorithm? Random forest algorithm is like several decision trees. So what is happening here is that several decision trees are running one after another, and the result, maybe it will get one gene multiple times. So it, one point of time it found a gene, and then because it is randomly initialized, maybe again it came to the same gene and found the same result. So this will run for a multiple times, and based on this we will be able to find which genes are coming most of the time. So that is called voting. So it sort of does a voting also. After running all these things, it will do a voting and give you the most stable and the most important set of genes which are coming over and over again. So we saw also the results last time, that we get what kind of results we get, like these sort of results. So this is an Excel sheet, uh, but this is the result we got, right? We saw this. So for today, let's just quickly run this so that you get more comfortable, and then understand why this gene is important, why this gene is important. So there are two features, two aspects here. One is accuracy, another is gene. So the accuracy tells you that the first gene that is there, it is there. If I remove this gene from the analysis, the accuracy will decrease. So this is the most important gene. The first gene is the most important gene, which is making this analysis this accurate. If you remove this gene, the accuracy of the analysis will go down. This is the kind of and in Gini talks about the stability. So in also in Gini you see that the same gene is there also. So top 4 genes are those genes which are really critical, because it is coming in both the results. So what is interesting for us is is these genes. So this is the output that I will be, gaining out of running random forest. And then I will look into these genes in more detail. So the more the genes, the better it is for me, because these pathways that we wanted to look into, these pathways are, these pathways contains 20 to 200 genes. So if you have like only 20 genes, then it will be, sometimes it's difficult to map the pathway. So we want to have like a lot of genes. That also helps. So this is the sort of output that we get after running, random forest. And uh, and uh, we this is the result, the output that we get. And when I open this in a, Excel sheet, and if I see, if we get like those 15 genes, and if I see the differentially expression of those genes, we will see that significantly some of them are different based on our different samples. So this sort of analysis we can do downstream. So you got all those 15 genes, you found out that 15 genes are there. From machine learning you got 15 genes or 20 genes or 100 genes. What you can do next, obviously you can read more about it and do all those things, but you also want to look at their gene expression, that how different they are, and what is there in these genes. Why, maybe you need to ask that question, that maybe I'll want to go back and see the differential, the expression of gene, because the data set have it, like, right? You are running the RNA-seq pipeline, you have the differential data, so you can plot it and see that whether, so this second graph is more relevant. Here it is all over the place. So here you can see that we have colored it based on the samples, like basal or normal, like an Claudin low. So this is just an example of and this file is also there with you, so that you can learn and do that. So all the files are here. uh, so we have sent you the link. So we have, you can, when you open, you will see these files. So this file is the one which is the Excel sheet, which is already being emailed to you. And the supervised machine learning pipelines that we run for 7,000 genes. So this is the train and test data for 15 genes and 7,000 genes. So 7,000 genes is the complete data set, and 15 genes is the small data set. And this is the output also for different algorithms. So this all is already been emailed to you. You can download these and look into the results and come up with questions or whatever, understanding or anything, like you can post it in the forum. But to do this analysis on your own, you can also run these pipelines. So how can we do that? We can do that using the T-Bioinfo platform. So because you have the files, you can totally go in here and easily run this analysis by yourself, because that is the whole purpose of this program, so that you can do it by yourself as well. So in here I will go into the supervised analysis. uh, so this is the supervised analysis window. Here you can see that you have several algorithms. You have random forest multi-run, random forest multi-run iterative. So what is random forest multi-run and multi-run iterative is what is that random forest, we want to run them several times. So when you have a like a data set, and when you want to expand to 15 genes to 200 genes, and you want to see that which pathways they are affecting, and you are doing all sort of biological analysis and you want more information, then you try these several methods, and then you find out more genes. By, if it is not coming by this method, I will try that method, that sort of thing. So you have several methods to test your data, test your understanding, test your analysis. That's why the platform provides several methods to do one kind of analysis. So if we, how we do this, we will have to upload file. And we learned this, that in supervised analysis, we have to give the machine two files. One is train, another is test data set. So I will here add files. So this is the folder, supervised files. I will go in that supervised files. I have 7,000 genes wala file and also 15 genes, and both have test and train data set. So both have test and train data set. So I will select both, and I will upload them both. So when you select both, both of them have been now uploaded. Okay. Now what I have to do, I have to just do group selection. Group selection is important because you have to tell which is train and which is test. So although we have named it like this, so I will have to drag and drop it here, train, and drag and drop it here as test. So I'm telling the machine that this is our train data set, this is our test data set. Now I will continue. Now I will push start button, and I will push OK. And then what we did, random forest. Okay, we did a random decision tree as well. So there is no parameter here. So okay, save, and then end, uh, save. So we did a decision tree for 15 genes. uh, okay, this is enough. Save, and we run the pipeline. So this is one pipeline is running for decision trees, and we will get the results afterwards. But we already have the results because I have given it to you already. But you can run this and you can do the same thing as well, because all of you have the access and the files also. So again, we will do the same thing to do another algorithm. We will just upload the files, and once it is uploaded, we will do the group selection, and we will put the test in the test dabba and train in the train one, and then we will continue and start. Now we can do random forest. So it is asking for how many trees do you want to do. So 500 trees, like this, 500 trees. So fine, okay, I will run 500 trees. And so random forest, uh, it is 15 genes. So there is also a data set that has 7,000 genes. You can do comparative. You can run 7,000 and you can run 15 genes. Like all of these analysis for 7,000, all of these analysis for 15. And then you can see what kind of results that you're getting, which one is better, why 15 genes. All those questions can be answered once you do it. So random forest, 15 genes and 500 steps I took. Okay. So save, run pipeline. So you can run these pipelines like this, and let's just check out one of the results. So let's check out the results from the data set that I have already emailed you. So let's look at the output. So random forest, I have 7,000 genes, 7K is 7,000, and 15, 15 genes also. So let's set 7,000. So we have confusion matrix. We get a result like confusion matrix, feature important. So this is the one with the genes which we were seeing. So let's open it with Excel. So here you can see accuracy and gening. So based on this, you can select those number of genes which are important. So this will give you 10,000 genes, 7,000 genes ka both the accuracy and from there you can plot this, you can filter this and found out the one which is most important based on the accuracy parameter here. So this is one way. So if I see for the smaller data set, the one that which I showed you was for the 15 genes. So let's look at that one also. And also confusion matrix gives you like the prediction results. So confusion matrix is nothing to get confused about. So it is read like this. Okay, so it has 4 groups: basal, claudinal, luminal, normal like, and error. So the last row is error. So basal, there are out of 15 genes, basal, let me just, you know, just for your visualization, improve this. This is normal light, 0, luminal is here, 0 is here. So in our samples, it predicted that, you know, the algorithm predicted that there are no basal samples. So it did not predicted any basal, 0, 0, 0, 0. But it predicted 4 Claudin low. And it predicted 20, 20 luminal. It will come up here, as you see. So it predicted in our test data set, there are 20, 20 luminal. Because we have not told the machine which is which, but it is predicting that which is basal, which is luminal. So this is the confusion matrix, which gives you, like, if you see like this, 20, and this has 2 normal, like, and it also gives you that if there is a false prediction. So that's why the accuracy, because we also tell them the test data set also have this parameter which tells them that which one is which, that is also there. So what the machine does is gives you the idea that how correctly it has classified. So one of the normal, like, was predicted as basal. One of the normal, like, sample is predicted as basal. That's why this is the error, that is the false prediction. So 0.033. So this is the confusion matrix, and we can discuss more in this if you have more questions next time we come up. But here are the set of genes. So this is the set of genes that you got after doing the analysis. So once you search about these genes, you will learn more about it. So that's the machine learning part that we kind of covered in this session, where we have these sort of information that we get. So if you have any questions, please ask. And next, what we do is biological interpretation. So there is this website or a database called David. So in here, what we do is gives the information as gene symbol. So there is a pipeline called annotation. Okay, so this file is also provided here. uh, Annotation 15 genes. So this is just the ensemble ID. So the ensemble ID of those 15 genes are there in this file. So in the pipeline here, you just have to input these IDs, and the output will give you the gene symbols, means the genes that are associated with this ID. And then you can give that input in David and find out about these different genes that are associated with this ID. So it sort of gives you a file conversion and finds out the genes that are associated with it for you to do the downstream analysis. So this is a part of interpretation. Similarly, we once if you remember, we did this pipeline before. We did that PCR clean and then expression, alignment to the transcript, expression table, and then differentially expressed genes. So we saw those differentially expressed genes, their fold change. So all many of these pipelines, like the GAGE, GAGE is another website which uses in which you have to provide the log to fold change. The input for that GAGE is log to fold change. So you have to provide these parameters which you get after DSEC. So after running DSEC, you can put this and do gene set or pathway enrichment analysis, and you will get a output like this. So this is a really nice output, because here you are looking at pathway, the complete pathway. So you're looking at regulation of actin cytoskeleton, you're looking at B cell receptor signaling pathways, and in here what you are finding is the gene expression. So if you see the colors, in this pathway, actually you're looking which genes are overly expressed, which genes are low expressed, high expressed. You can see this, and you can design a hypothesis that why this disease and all those things, all the mechanistic details based on this information. So this is a very critical and important sort of a result that that a researcher can get, and to do more further analysis, because this will tell you the whole story. So human gauge is also there. So I will just quickly show you also the files. I have given you the files here. So for human gauge, the files are given to you, dsec output or else also hrc output. I have put it here, so you can upload these files and run the human gauge. And I'll just quickly show you where it is. So this our random forest is done. Quickly look into the result. So you have confusion matrix, features, and this test data set. And this is the R package, pura R package that you can import into R and do other analysis. So you will look into feature importance. So this is the important file, and also the confusion matrix you can see. So this is done. I think you will be able to do this easily now. If not, you can always ask us how to do it. So I wanted to show you the utilities. So in utilities, when you go, you will find this annotation, human gauge. So these two you can run and do the biological interpretation. So coming back to the presentation, this is how you can learn to do human gauge, and then look into the critical pathways. So we finished sort of a lot of things. So I would really want you guys to go through transcriptomics 1 and 2 and 3 slowly, but at least start doing it. I mean, it's not so difficult, and I mean it's in there in your the program fees that you have paid. So it would be nice, I mean, to explore these are like nothing but text that you have to go through, some of the hands on. So it tells you like how to do everything, like everything. So it's like fully independent. So you don't have to depend on any session or anything. It starts from the basic and goes all the things that we described here. And also get like certificates, like these are really valid certificates and that you can showcase all the time in your career. So I mean, get those certificates. There are 3-4 certificates out there if you just finish the courses. So I think that would be a good thing for everyone here. So in the next session, we have the projects, which is on 27th. So it would be nice, like if you can finish this, then we can actually plan for these projects that are there in the platform. But without doing this, doing a project will be different. So in case you are not at all being able to do this, then the project would be to complete these courses, because once you do that, you will be more and more comfortable with whatever we covered today, and also what we talked and all been talking about. So once you do this, you will be an expert in yourself as well. So another thing that I wanted to say is that day after tomorrow, on Wednesday, we have this free webinar. And it would be really a great webinar, because we will be talking about the Africa program and then precision oncology. Like, because we talked about precision medicine today, like in that we will be talking in more detail that how oncology problems have been handled, not by looking into the transcriptomics data, but also the genomic data, also the other kind of data, and how these problems have been solved in oncology, and how the research is ongoing. We will learn about cancer, we will learn about the basic cancer, and then we will learn about genomics, then we will again go through about transcriptomics, and then we will go into these different data sets. We'll go into ncbi, like you saw. tcga is another very useful data set. Cosmic, where it gives you all the mutations that are there in the gene. So this is a 2.5 month program which will run and do all those things, which is bioinformatics for precision oncology. But on day after tomorrow, we have this free webinar at 9:30 p.m. Indian Standard Time. So if you have time on Wednesday, please join us for this free webinar, and we will be sharing the link of that Zoom link to all of you as well. And if you have any questions, like put it there, put in the forum, and wherever you're comfortable, WhatsApp group, and we will be helping you with those. As I was telling you, on 27th we have this great session where we would show you that kind of amazing projects that we have, and that you can work, and you can put it as your experience, and also it will be there in your certificate. So it would be really awesome. So if you can finish some of the materials that we discussed today. And I think with this I will conclude today's class. And if you have any questions, please ask us by any of those means. And I hope that this was a useful class for everyone, and I'm open for your feedback and suggestions. Thank you.